Okay, here we are back for the second video lecture for Chapter 9 on cellular respiration. You'll recall that in the first video lecture for this chapter, we had talked very generally about cell respiration being a mining process. In other words, mining our food for its electron content because the electrons represent the energy of our food. And so we basically said that we need to be able to get the electrons out of the food molecules by breaking them down and then transport those electrons from one molecule to the next via redox reactions until finally they can be sort of offloaded at the electron transport chain, which is this last figure here that we talked about in the first video. The electron transport chains, again, are a way to extract the energy from those electrons in sort of a slow, step-by-step, piecemeal fashion so that it can be, or the energy release can be controlled, which means you get more energy out that you can use. It makes the process more efficient. Okay, what we're going to start talking about in the second video, though, are the major steps in the process of cellular respiration. All right, so we have really overall three major steps in our process here. The first one is called glycolysis or glycolysis. Remember, the term lysis means to tear, rupture, or break apart. Glyco refers to sugar. So this term means sugar splitting, okay? Breaking down a molecule of glucose into two molecules of a 3-carbon sugar called pyruvate. We'll see more about that here in a little bit. The second step is called the citric acid cycle. You may also see this referred to as the Krebs cycle, okay? But citric acid cycle is what the term I generally use, and I think it's the more common term, but you might see it expressed both ways, and that's fine. The citric acid cycle is sort of like a grinding mill. It's going to grind down the organic sugar molecules and extract the remaining electrons from them, okay? Now that extraction process begins in glycolysis when glucose is broken down into two molecules of what's called pyruvate, but the citric acid cycle completes the grinding down or the breakdown of those sugar molecules to extract the remaining electrons. And then third, we have what I referred to sort of generally in the previous video as the power plant step. This is known as oxidative phosphorylation. Okay, oxidative phosphorylation. This is where we're going to get lots of ATP produced. Okay, so that's a really important step there. It's going to produce a large amount of ATP. And I'll probably cover that in a third video lecture for this chapter. Okay, so let's see how all this kind of breaks down, or sort of a summary of this. Okay, so first of all, what we have is our mitochondria. You can see that here, all right? Now, glycolysis, our first step, happens in the cytoplasm of the cell. It does not happen in the mitochondria. Okay, it takes place outside the mitochondria in the cytoplasm of the cell, and it's going to break a glucose molecule in two. So glucose has 6 carbons, right? C6H12O6. If you sort of snap that in two through the process of glycolysis, you end up with 2 molecules of a 3-carbon sugar called pyruvate. So take a 6-carbon molecule, snap it in two, you get 2 3-carbon molecules. They're called pyruvate. Now, this process does produce a small amount of ATP, about 2 ATPs, okay? So plus 2 ATPs. That's a process called substrate level phosphorylation, and we'll see how that happens when we talk about or get into glycolysis. But the really important aspect of glycolysis is that this is the first step of the electron mining process. So electrons are going to be loaded into NAD+. Remember, we have NAD+ as our oxidized form, and it'll be reduced to NADH as electrons are loaded into it. And so we have electrons coming out of the process of glycolysis, and they're being shunted over to our power plant step, where the electron transport chains are, all right? Okay. Then, at the end of glycolysis, we have our 2 molecules of pyruvate. They will enter the mitochondrion, but there's a problem here. The pyruvate is a molecule that's not compatible with our citric acid cycle that you see over here. This is the grinding mill I talked about, okay? So what needs to happen is that the pyruvate, once it goes into the mitochondria, needs to be converted to a fuel that is compatible with the citric acid cycle. That fuel is called acetyl-CoA. That is short for acetyl coenzyme A, all right? So this is just this reaction here called pyruvate oxidation, or also called the PrEP reaction, converts pyruvate to acetyl-CoA, which is now compatible with the citric acid cycle. All right, that's a simple reaction. We'll see that in a little bit. Okay, then the acetyl-CoA drops into the citric acid cycle, and basically the cycle breaks this acetyl-CoA all the way down. So what's going on here is that we have glucose, or organic carbon, I should say, as glucose, ultimately being broken down and given off as CO2 by the citric acid cycle. So we have organic carbon going to inorganic carbon. That's a breakdown process. It's catabolic. And you can see that there are a large amount of electrons coming out via NADH, but also now through a second electron carrier molecule. Just think of it as a different colored electron bucket if you want. FADH2. These are both carrying electrons now over to the power plant step, oxidative phosphorylation, where the electron transport chains are. Now there's a small amount of ATP again made here in the citric acid cycle through the same process called substrate level phosphorylation that we saw in glycolysis, but again just makes 2 ATPs. I mean, they contribute to the total, but it's not a whole lot. Look at all this ATP that we get here. Typically we get, I don't know, about 36 ATPs, something like that, in oxidative phosphorylation. And again, this is where the oxygen is involved in pulling electrons down the transport chain. So you can see now why it is that breathing oxygen is so important. If you can breathe in oxygen and use it in oxidative phosphorylation, you can make a large amount of ATP, much more so than organisms that live in anoxic environments, or environments where there is no oxygen. Okay, that's a brief rundown of the process. Now we're going to dive into each of these steps individually. Okay, so basically, as I said, we have a lot of ATP being made in oxidative phosphorylation. Typically, it's about 90% of all the ATP made by this entire process. 90% is made in oxidative phosphorylation, okay? Smaller amounts again are made in the citric acid cycle and glycolysis through substrate level phosphorylation. Okay, so for each molecule of glucose that's broken down to CO2 and water by cell respiration, the cell makes, well, the number varies, anywhere from maybe 32 to 40. It varies a little bit here and there depending, but 32 to 40 ATPs. It's quite a bit. Okay, let's see how generally substrate level phosphorylation works. Now this is again the process of making ATP that we see in glycolysis and in the citric acid cycle. So substrate level, think about what a substrate is. A substrate is a molecule that an enzyme operates on, right? We've talked about that. So we have an enzyme molecule, protein, that we see here in purple, and it'll have an active site that accommodates an organic molecule that has phosphate groups on it, and an ADP molecule, adenosine diphosphate, okay, 2 phosphate groups on that molecule. So this substrate molecule is a high energy molecule. You know that because it has a phosphate group attached to it. Well, if that substrate molecule fits into the active site along with an ADP, the enzyme will break this bond connecting the phosphate to the substrate and then attach that phosphate group, that high energy phosphate, to ADP, making ATP. It's sort of like picking apples off of trees, right? You pluck an apple off of a tree, now you've got a high energy food source. So plucking phosphate groups off of substrates, organic substrate molecules, and attaching to ADP, now you've got ATP. That's the general process of substrate level phosphorylation. It's more complicated than that, but that's a pretty good sort of broad overview of it. Okay, so now let's talk about glycolysis, which is our first step, right? This is the first step of the mining process that we call cellular respiration. Glycolysis, again, means sugar splitting. is going to break glucose down into 2 molecules of pyruvate. So we have glucose going to 2 molecules of what's called pyruvate. Pyruvate is a 3-carbon sugar, glucose is a 6-carbon sugar. Now, there are 2 steps, or 2, I guess, sections or phases, maybe I should say, of this process. First, we have what's called the energy investment phase. Energy investment phase. We need to be able to inject a little bit of energy into this process to kickstart the process. So we talked about energy of activation back in chapter 8. We were talking about proteins, if you remember, and how they act as catalysts. They lower the energy of activation. Well, even a catabolic, spontaneous, exergonic process requires energy of activation to kickstart the process, okay? So this energy investment phase is where in glycolysis a small amount of energy is added to glucose to make it so unstable it then begins to break down. Again, this is like putting a flame to the house, right? You're adding activation energy to kickstart that process. Okay, then once the reaction begins to move forward, we enter what's called the energy payoff phase. This is where we get more energy out than we invested in the investment phase. So it makes the overall process a net plus in terms of energy, okay? It makes energy available for the cell. Now, glycolysis, a sort of unique thing about it is that it does not require oxygen. Glycolysis can happen whether O2 is present or not. This is one reason why we suspect that glycolysis evolved very early on in the history of life on the planet, well back before there was ever any oxygen in the atmosphere that organisms could have used. Okay, so it's a very ancient process. So let's take a look and see where we are in the scheme of things here. All right, so again, this is sort of a graphical representation of the major steps in cell respiration. We are here at the very beginning in glycolysis, okay? All right, so let's look at a breakdown of the overall process here. Okay, this is glycolysis. So what we see in the top right here is the energy investment phase, okay? So how do we invest this energy? In what form? 2 molecules of ATP are going to supply the investment energy we need to kickstart this process. So 2 ATPs are used, phosphate groups are split off of each one, the phosphates are added to the glucose molecule at various steps along the way, and they make it really unstable so that it begins to break down. Well, when you pluck 2 phosphates off of 2 ATPs, what do you get? 2 ADPs. So this is ATP breaking down, giving off energy. That energy is being applied to glucose to make the reaction go forward. That's our energy investment phase. So we've expended already 2 ATPs. We're sort of in the hole, right? We've gone to the negative side of the ledger, if you want to think of it that way, in terms of energy. But that's okay because we still have the payoff phase coming up. In the payoff phase, as the reaction goes forward, enough energy is given off to add 4 phosphates to 4 ADPs, making 4 molecules of ATP. So we use 2 in the investment phase, but we get 4 in the payoff phase. What is our net profit? Plus 2 ATP. So 100%, right? We've spent 2, but made 4. We have 100% on our money, so to speak, here. Okay. Now, here we see glucose breaking down to pyruvate, right? We talked about the ATP being formed, more formed than we used, so it's a net plus for us. But this is what's really important here, the electrons coming out. So you see we have 2 molecules of NAD plus coming in, scooping up 4 electrons, and remember, protons are always following them, right? And reducing NAD plus to NADH. Now we have a high energy electron carrier molecule, and NADH will carry those electrons over to our transport chains, where their energy will be extracted and used to make a lot of ATP. So this is sort of a net summary of the overall process. Oops, sorry, we'll go back. This is a net summary of the overall process. So a glucose is broken down into 2 pyruvates and produces 2 waters. we have 4 ATP formed, minus the 2 we use in the investment phase, so we net out 2. We're ahead of the game now. And importantly, we have 2 NAD plus being reduced to 2 NADH. Okay, so this is a broad overview of the process. It's important to understand big picture of what's going on with glycolysis before we get into the details, which are the next few figures. Now, these next couple of figures, they're a bit intimidating. There's a lot going on, but I'm going to water it down, and I want you to focus just on the stuff that I talk about, okay? So there are a lot of figures, for example. There are a lot of molecular structures. You don't need to know those. You don't need to know the names of any molecules unless I specifically point them out. I want you to think about the overall process. What is going on overall, okay? All right, so this is a diagram then of the energy investment phase of glycolysis. Okay, so we start off with our glucose molecule right here. So glucose is shown as the ring structure, again, is C6H12O6. Okay, first thing that happens is we have one molecule of ATP coming in and being broken down to ADP. So where'd that phosphate go? Right there. There you see it. Here we have a phosphate, here we don't. Now we have a new sort of form of glucose. It's called glucose 6-phosphate, but don't really worry about that too much. This is an intermediate, right? This is a biochemical pathway, and all the things that are formed between the initial reactant and the final product, again, are called intermediates. Okay, so this is a higher energy molecule than glucose was, because you know that because there's a phosphate group attached to it. ATP has supplied energy in the form of a phosphate. The next step is just a rearrangement. So this molecule is rearranged into this molecule. Again, the name of it is not that important. That sets the stage for our second molecule of ATP to come in and donate a second phosphate, and now you see it. Now we have 2 phosphates on what used to be glucose. This molecule you should know the name of, fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. So because it has these 2 phosphates on it, it's extremely unstable, okay, very unstable. So let's just write that down. Very unstable because it has a lot of energy in it via those 2 phosphates. So as soon as it's formed, it essentially snaps in 2. Because it's extremely unstable, it snaps into the 2 halves of it become these 2 molecules. One half of it becomes what's called G3P, glyceraldehyde 3-phosphate. You should know that, G3P. The other half becomes what's called DHAP, dihydroxyacetone phosphate. You should know that one too. So think of fructose 1,6-bisphosphate as a stick. You grab it by both ends, you snap it in 2, you have your 2 halves, right? One is called G3P, one's called DHAP. But there's a problem here. The problem is that we now need to go into the energy payoff phase, the second half of glycolysis, but only G3P is compatible with the second phase. DHAP is not compatible. So if there's not some way to fix this problem, right there we've lost 50% of the energy that we have here in these 2 molecules, in the sense that 50% of the energy is tied up into a dead end. It's tied up into a molecule that can't be used in the energy payoff phase. But there's a fix for this. We have an enzyme called isomerase. You see it right there. So remember, enzymes are named for what they do. Isomerase is going to convert DHAP into G3P. So these 2 molecules, DHAP and G3P, are isomers of each other. Isomerase converts DHAP into G3P so that we do actually end up with 2 copies of G3P for every glucose we started with. And those 2 copies of G3P then get funneled into the energy payoff phase, which is next. All right, so now let's follow these 2 G3Ps through the payoff phase. Okay, here's the energy payoff phase. So notice we have our G3P here. They're just showing 1. I don't know why they do that. They're just showing 1. They should show 2. But also notice that on all these intermediates, you have 2 panels. You see one in the front and one in the back, one in the front and one in the back. That's showing you that this process is happening twice. Why is it happening twice? Because we have two G3Ps dropping into this, okay? So instead of just showing this, here and then maybe again down here, which would be a complicated figure, they've shown it as a superscript 2 and two copies of each of these molecules, okay? Because it's happening twice, once for each G3P. Okay, let's follow this through. So we have our G3P here, okay? Now, this first step of the energy payoff phase is really important, okay? When G3P is broken down into, or converted rather, to this intermediate, that process is highly exothermic. Okay, what does that mean? It means that reaction gives off a lot of energy in the form of heat. All right, so we have electrons coming out, NAD plus scooping up electrons and being reduced to NADH, okay? And that allows that reaction to go forward. A lot of heat is produced, and so what happens? There are inorganic phosphates, P sub I, inorganic phosphate groups floating around. Think of them as like soup ladles. They're going to scoop up some of that released heat energy. They become energized, and they attach themselves onto G3P and convert it into our next intermediate. Notice G3P had one phosphate. Now we have a second phosphate, okay? Why two phosphates? Because we have two copies of this molecule. Each of them gets its own phosphate. All right, well, why does this happen? I mean, why are our phosphates being energized and stuck back onto these molecules again? Because this is the energy payoff phase and we need to get ATP out. How many copies? 4. So we need to replace the 2 we spent in the investment phase plus make 2 more to make this profitable, right? Well, or to make ATP through substrate level phosphorylation, which is what all of this is, you need to have high energy phosphates. These are low energy phosphates. When they scoop up that heat energy, they become energized, they attach themselves to their intermediates, and now we have apples on our tree. We have phosphates that can be plucked off and added to ADP. And that's what this next step is. This is really our first step of substrate level phosphorylation right here. So now we have high energy phosphates. We have 2 ADPs coming in. Each of them plucks off 1 ATP. So we form 1 phosphate group. So we form 2 ATPs. And now we have this intermediate, 2 of them, 2 copies, 1 in the front, 1 in the back. Each has a phosphate. We still have 2 more phosphates we can pluck off and make 2 more ATPs with for a later step of substrate level phosphorylation. All right, this step and this step are really just rearrangements of matter. They're not that important, okay? Don't worry about the names, don't worry about what's going on here. But ultimately, we get to this intermediate, which is the beginning of our last stage or step of substrate level phosphorylation in glycolysis. We have 2 more high energy phosphates, 1 on each of the 2 copies of this. 2 ADPs come in, pluck them off, convert to 2 ATPs, and now we have our 4 ATPs total through this process. That last step right there, that last substrate level phosphorylation step here, gives us our 2 pyruvates. And the 2 pyruvates are what we end up with at the end of glycolysis, okay? All right. So whereas it's easy to say that glycolysis is just taking glucose and snapping it in 2, and the 2 has being pyruvate, you now see there are a lot of steps involved in that process, okay? All right, but certainly the substrate level phosphorylation steps are very important, as are the electrons coming out and being loaded into NADH. Okay, so now we have the original sugar glucose converted to a new sugar pyruvate. We still have a lot of potential energy in the pyruvate that can be used. So this is where the pyruvate is then going to enter the mitochondria. Remember, glycolysis we've talked about here happens in the cytoplasm, okay? So the thing is that there's sort of a molecular fork in the road here, okay, in terms of what's happening at the end of glycolysis when we get our pyruvate. If oxygen is present in the cell, then that pyruvate will enter the mitochondria and all the rest of the stuff that we're going to talk about will happen. Lots of ATP produced. But what if there's no oxygen present, rather? If there's no oxygen present, then the pyruvate does not enter the mitochondria, and instead a process called fermentation takes over. Okay? Fermentation is a way to break down sugar molecules in the absence of oxygen. Now, it only makes a net 2 ATP molecules, okay? So fermentation is basically glycolysis plus a few reactions afterwards, but these ATPs that you see made here during glycolysis, here and here, they are the only ATPs that are made during fermentation. So if you've got a choice, you want to run aerobic respiration with oxygen present, because the power plant step, oxidative phosphorylation, makes a lot of ATP. If you don't have a choice, fermentation is what you got to deal with. If you live in an anoxic environment where there's no oxygen, fermentation is what you have to do. A lot of bacteria, fungi, things like that will run fermentation because they really don't have any choice. So they live in an anoxic environment. Think about the sediments at the bottom of a lake or the ocean or a wetland or something like that, or in the gut of an organism, right? So fermentation is an old process. It evolved before oxygen began to develop in the atmosphere around 2.5 billion years ago. It was present long before that, and it works because it doesn't require oxygen. Once oxygen began to build up in the atmosphere around 2.5 billion years ago, once photosynthesis kicked in on the planet, then we had the evolution of the next stages of cell respiration, citric acid cycle, oxidative phosphorylation, that we're about to talk about. Okay, so let's assume we have oxygen present. The pyruvate then is going to enter the mitochondria, and then, like I said, we have a process called the PrEP reaction. Okay, the PrEP reaction, which is going to convert the pyruvate that we ended up with at the end of glycolysis into what's called acetyl-CoA, or acetyl coenzyme A. All right. So this is going to convert pyruvate into a fuel, acetyl-CoA, that's then compatible with the citric acid cycle. All right. Okay, so let's orient ourselves and see where we are. Okay, so we've got our pyruvate, and now it's going to enter the mitochondria, and our PrEP reaction occurs. Technically, the PrEP reaction is called pyruvate oxidation, but everybody just calls it the PrEP reaction. Okay. All right, so let's see what's going on in the PrEP reaction. This is it. Okay, so we got our pyruvate. This is our pyruvate molecule right here. All right, now notice that we have 3 carbons, right? And there are going to be 2 copies of pyruvate. Think about it. We start off with a 6-carbon glucose molecule. You snap it in 2, you end up with 2 3-carbon molecules called pyruvate. So we still have all 6 of our original organic carbons that we had in glucose. It's just now they're separated into 2 molecules of 3-carbon pyruvate. Okay. Pyruvate enters the mitochondria through a membrane protein, a transport protein. Goes into the interior of the mitochondrion, and the first thing that happens is that this portion of the pyruvate molecule breaks off and becomes CO2. All right. That CO2 represents 1/3 of the CO2 that comes out of you every time you exhale. Okay, 1/3 of it is produced right there. All right. Second thing that happens is that we have more NAD+ coming in, scooping up electrons and being reduced to NADH. And those electrons will go over to our power plant step. Third thing that happens is that a molecule called coenzyme A attaches to the second carbon where the CO2 used to be. And now we have acetyl-CoA. Now we have something that is compatible with the citric acid cycle. Okay, now our fuel is compatible with the citric acid cycle. All right, so the citric acid cycle is something we're going to talk about really in more detail in a minute, but for now I just want you to think of it as being a complicated process that we're going to kind of gloss over a little bit, okay? Citric acid cycle basically is going to take that carbon and break it down the rest of the way into CO2. Okay, it's going to take that organic carbon, these two that we see here, these two in red, for example, okay, and there are 4 of those because we have 2 copies of acetyl-CoA now. The citric acid cycle is going to convert those organic carbons into inorganic form as CO2. Okay, so 2/3 of the CO2 that you exhale every time you breathe out comes from the citric acid cycle, converting these organic carbons into CO2. All right, so let's talk about the citric acid cycle then. Well, basically what it's going to do here is oxidize acetyl-CoA. So think about redox. Oxidation is loss of electrons, right? So again, this is more of the mining process. If the citric acid cycle oxidizes acetyl-CoA, it's extracting electrons from it. And those electrons then have their energy sort of siphoned off by the transport chains of the power plant step, which we call oxidative phosphorylation. So the citric acid cycle is called a cycle because it begins with a certain molecule. Okay, it begins with a certain molecule, and that molecule is basically converted to various intermediates, but then you get that molecule back again at the end of the cycle. That's why it's called a cycle. You start and end with the same thing, the same molecule. Okay, the cycle is going to oxidize acetyl-CoA. It's going to generate 1 molecule of ATP. So 1 molecule of ATP, so plus 1 ATP, is going to produce 3 molecules of NADH and 1 molecule of another electron carrier molecule called FADH2. So this is what the citric acid cycle is going to produce, okay, per turn of the cycle. All right, so let's take a look at that. Okay, so here at the top we have our prep reaction. We just talked about this, right? We have pyruvate being converted to acetyl-CoA, okay? All right, the acetyl-CoA is going to drop into the citric acid cycle, and the cycle is shown sort of as a circle here because there's a molecule here we'll talk about in a little bit that picks up that acetyl-CoA. When the acetyl-CoA comes in, the coenzyme A portion breaks away, and think of this as injecting those 2 organic carbons into that initial molecule, that starting molecule, or what I call the gateway molecule because it's the gateway to the cycle. A series of reactions occur. We get these 2 carbons ultimately coming off as CO2. Okay, we get electrons coming out as NADH in a couple of different places, one here and then as FADH2 here. And we have some substrate level phosphorylation here, just like, or very similar to the way it happens in glycolysis, all right? So this is just a very broad summary overview of the process. It's more complicated than this. There are basically 8 steps to the citric acid cycle, and each one is catalyzed by its own specific enzyme, so it's a metabolic pathway, okay? The acetyl group of acetyl-CoA joins the cycle by combining with a gateway molecule called oxaloacetate. So that molecule is located right there. Oxaloacetate, that's our gateway molecule. You should know that one, all right? You should know the name of that molecule. Okay, so oxaloacetate is the acceptor for the acetyl coenzyme A, all right? Now, it accepts these 2 organic carbons, in other words. All right. Over the course of this process, that molecule is converted to various other intermediates, and then the back half of the process rebuilds oxaloacetate. So it's there for the next acetyl-CoA molecule to drop in. So we start with oxaloacetate and we end up again at the end of this with oxaloacetate. That's why it's called a cycle. All right. The NADH, the FADH2 transport their electrons to the power plant step of oxidative phosphorylation. So again, just to orient you, here we are now with citric acid cycle, all right? Okay, so citric acid cycle is complicated, all right? But as I did with glycolysis, I'm going to kind of water it down. There are a lot of structures shown. You don't need to know those. There are a lot of names of molecules. Unless I say you need to worry about them, you don't need to worry about them. All right? Okay, so let's see what's going on here. All right, so this is the citric acid cycle. We're going to start here at step 1. There again, you see oxaloacetate. Okay, acetyl-CoA is going to join up with oxaloacetate. The acetyl group, which you see here with the 2 carbons in red, will attach onto this right there. And now we get our first intermediate. You should know this one. It's called citrate. That first organic intermediate, called citrate, or also citric acid, is why the cycle gets its name. It's why it's called citric acid cycle. But notice that it is a 6-carbon compound. 1, 2, 3, 4, 5, 6. Keep track of these shown in red, okay? They're the ones that came in as acetyl-CoA. All right, step 2, we have, basically a rearrangement into this intermediate. Don't worry about the name. You can still see we have our 2 carbons from acetyl-CoA right there. Third step, this is our first electron mining step of the cycle. NAD plus comes in, is reduced to NADH, all right? This is also where we get the first CO2 produced of the cycle. And notice that that carbon in that CO2 is blue. That's because it's one of these 2. It's not one of the 2 that came in at the beginning of this cycle. It's a carbon that was already present in oxaloacetate. These carbons drop in as acetyl-CoA, and they spin a turn or 2 in the cycle before they eventually come off as CO2. Okay, they don't come off as CO2 in the same cycle in which they drop in. They come off as CO2 in a cycle or 2 later. All right, that process converts this intermediate into this one. Again, don't worry about the name, not important. Fourth step, this intermediate is converted to this one, and this is where we have our second round of electron mining. NAD plus comes in again, gets reduced to NADH, and we get a second CO2. So by this point, all of the organic carbons that used to be present in glucose at the very beginning of all this have now been converted to CO2. This is essentially the end of the grinding process that breaks that organic carbon molecule down into CO2, okay? All right, these electrons again, we'll go over to our power plant step. Okay, now we have our substrate level phosphorylation step here. Okay, first of all, don't worry about this GTP, GDP. Okay, just think of them as similar to ATP and ADP. So we have this reaction here, which is exothermic. It gives off energy. Phosphates absorb some of that energy like we saw before, and they get attached ultimately to ADP, converting it to ATP. That's all you need to know about this substrate level phosphorylation step, okay? That's going to make 2 ATPs total for every glucose we started with at the beginning of this process. All right, now we have this intermediate. Don't worry about the name, not important. But now in step 6, we have a molecule called FAD. Just think of it as another empty electron carrier molecule, oxidized electron carrier molecule, coming in, scooping up electrons and being reduced to FADH2. Those electrons go to the power plant step. All right. Now we have a few more steps here, and there, and so on. We have another electron carrier molecule, NAD plus, coming in, scooping up electrons, being reduced to NADH. That last round of electron mining rebuilds oxaloacetate. Really, the back half of this cycle is about regenerating oxaloacetate and in the process getting out more electrons, okay? All right, so at the end of all this then, we have a lot of potential energy in the form of NADH, right? And FADH2, and we've converted, or regenerated rather, our oxaloacetate molecule, okay? Plus, we've made 2 ATPs total. Remember, we have 1 ATP being produced here, but we have 2 acetyl-CoAs for every glucose we started with. So we get 2 ATPs for every glucose we started with in the citric acid cycle. Okay, so the next step then, the last step, is going to be oxidative phosphorylation. That's the power plant step, and we're going to save that for the third and final video lecture for chapter 9 here, okay?